The overall goal of this experimental procedure is to observe the cyclic AMP-dependent regulation of fibroblast migration by Mrp4.
High-content microscopy can help answer key questions in the field of cell biology. For example, in addition to conferring drug resistance, Mrp4 can also regulate physiological processes such as cell migration by modulating intracellular cyclic nucleotide concentration.
The main advantage of FRET imaging that we are going to attempt today is to allow monitoring of the intracellular dynamics of cyclic AMP and cyclic AMP-dependent kinase in a real-time fashion.
The implication of high-content microscopy is it can be used in drug discovery. Once the system is set up or the instrument is set up, there is no human intervention involved in this plate-based assay.
Though this method can provide insight into cell migration, it can be applied to other systems such as live cell non-perturbing high-content imaging of immune cell killing, clonal dilution, angiogenesis, cell proliferation, chemotaxis, and 3D spheroid.
To prepare cells for high-content microscopy, under a cell culture hood, use a 1% fibronectin solution in PBS to coat the wells of a 96-well plate. Incubate the plate in a CO2 incubator at 37 degrees Celsius for 2 hours. Aspirate the fibronectin solution from the wells. Then, after resuspending Mrp4 negative and Mrp4 positive cells according to the text protocol, use a hemocytometer to count the cells. Then, seed 30,000 cells into each